Is it recording?
Yes. Okay, very good. Welcome to Jessica Rubin's Masters in Science Defense. Joseph Gorez is her leading on her committee, as well as me. I'm Terry Delaney. I'm the chair of her committee. So I will let Joseph introduce Jess. Thank you, Terry. So welcome. You at home and everybody here in the room. I have the pleasure to maybe get somebody out of the door today, and it's good, Rubin. Jessica I've worked with Jessica quite a while ago, so she's not new to my lab. So we actually met through a project where we looked at earthworms, invasive earthworms in sugarbush. And that was about 5 or 6 years ago. And so I was kind of surprised when she turned up again. I obviously didn't turn her off or make her feel like uncomfortable. She turned up and said, I want to be that street. So you should know a few things about Jessica. First of all, she got her degree in 1998 in ecoliterature at Cornell. And then she got another degree, a master's degree in environmental studies and a certificate in science education from Antioch College in the northeast part of Antioch College. And in between 1998 to 2012, she did a whole bunch of other things. If you want to have any kind of advice on outdoor living, sleeping in the snow, tracking all sorts of animals, then Jessica's the person for you. But she came to me with this idea of looking at mycorrhizae as ways of mitigating phosphorus in riparian areas. And so over the time that she's been here with us, she was able to get $100,000 worth of money. Kind of looked at it this morning and saying, wow, $1,000, really, that's 4 grants, 4 or 5 grants. And she was able to get 3 of her papers accepted that are in her thesis. Today she'll be mainly presenting on one. And so one other thing that I want to say, she really opened my eyes towards Eoboniki. So Abenaki to some of you, Eoboniki here in Vermont, and their skin in this game, right? So we are on unceded territory, and it'll be really nice to rematriate some of the ecosystem services that are provided by these riparian folks. With that, I invite Jessica to come forward and give a talk.
Thank you so much, Joseph. Thank you everyone so much for being here today. And thank you everybody on Zoom. And I'm really grateful to be here. It's been an incredible privilege to get to be a student here at UVM in the Plant Soil Science Department, studying out of Dr. Joseph Gorez's lab and being invited to be part of the Hurley-Faulkner lab group. And I'm really grateful today to discuss some of my research exploring mycorrhizae and riparian restoration to enhance phosphorus mitigation and pollinator habitat on unceded territory. And I also would like to give thanks to my committee, Dr. Terry Delaney and Stephanie Hurley. So saying that we're on unceded territory is certainly not enough. You will hear more about that. How to advance the slide.
Screen sharing, so I'm going to undo that. It says resume in green above that.
And I'm trying to advance to the next slide. There we go. All right, so the outline: objectives, rematriation orientation, ecological restoration lens, literature review, and mycorrhizal ecology. I'm only going to be sharing about a small bit of my research today, case study at Shelburne Farms. And as Joseph mentioned, this will be coming out in Restoration Ecology Journal this spring. And we do have other mesocosm studies I will not be discussing that are coming out also this spring in a subjournal of New Phytologists called Plants, People, Planet. Be discussing acknowledgements and there'll be time for questions. So starting with the first objective of rematriation, for those of you who may not be familiar with the term, it refers to Indigenous people obtaining their birthright access to their Indigenous lands. And this was not one of our original objectives, but it evolved as like a necessary objective as the project developed. Our next objective of water quality protection through mycophytoremediation involves working with fungi and plants and pollinator habitat restoration. So I begin with this picture of Elder Mycorrhizae and Abenaki clan mothers guiding some of our community to a rematriation ceremony between the lake and our research site at Shelburne Farms, summer 2020, thanks to collaboration of Alno Byway, New England Grassroots Environmental Fund, and Myco Evolve. And this is sort of a lens through which I conduct this research in as a third generation American from Eastern Europe with the privilege of white skin and settler status. How do I attempt to restore ecosystems in a way that's not just another neo-colonial project? The field within which I'm presenting, ecological restoration, is a fairly new field in terms of being recognized in the scientific community. It's really only was honored by that community in the 1970s, and a lot of scientists take issue with the term. In this book, they discuss that reconciliation is a more apt term because technically you can't restore ecosystems to what they were, and to try to restore ecosystems to a situation before humans appeared is not realistic. So the term reconciliation more accurately describes that we have to reconcile humans are here, the damage we've done, and how we can change our ways. A subfield of that, mycorrhization, involves working with fungi, and another subfield, phytoremediation, works with plants. Together, mycophytoremediation works with fungi and plants. Formerly, I'd only seen that field applied to heavy metals. In this context, I'm applying it to nutrients. This diagram on the right demonstrates from a mycorrhizal ecology textbook, as you move from the molecular level to the ecosystemic level, not only does the picture become more blurry, but precision decreases. However, relevance increases. And this is another lens through which I conduct and offer this research of humility, because there's actually more we don't know than we do. So out of all the many types of fungi that I am working with, a particular category that was coined by German mycologist and plant pathologist Albert Frank in 1885 with the Greek root of the word myco, meaning fungi, and rhizae, meaning root. 2 years later, he coined the term symbiosis, and it's worth noting that mycorrhizae fungi have a spectrum from parasitism to mutual. For those of you not familiar with mycorrhizae fungi, they partner with 90% of plants. They play a key role in terrestrial ecosystems. They regulate nutrient cycles, influence soil structure, and contribute to ecosystem multifunctionality. So in the ecological restoration community currently, there are these 9 criteria that deem a site restored. And you can see that the first 4 refer to abiotic and biotic factors associated with plants, and the following 5 refer more to functional and developmental characteristics. And we put forth 2 additional criteria. The first being that the system satisfies the mitigation function. And the reason for this is because often ecological restoration projects will attempt to restore an ecosystem, but not actively address the pollutant. In our case, the pollutant is a macronutrient phosphorus that can serve as a pollutant when in water. The second criteria that we put forth is social injustice inherent in the environmental damage needs to be repaired. And what this means is that any research design needs to acknowledge the culture of the Indigenous people that the research is done on, integrate their expert knowledge, and reciprocate them with reparations that support the rematriation. And this falls within the 5 shifts paradigm of TRISOS, which emphasizes the importance of cultivating a decolonial ecological ethic. So the next objective, water quality, really stems from years ago when I was teaching earthcrafts at a Waldorf school in East Montpelier on a camping trip. One of our students playing in the Kingsbury Branch River ended up in the ICU a few days later with almost kidney failure. Luckily, he's thriving today, but it was frightening. And we tracked the strain of E. coli and it indirectly to cattle upstream. And it occurred to me that despite all the amazing water quality professionals that are both in this room and in the state and on this call, that there's still a lot more work to be done. This is not only just where we live, but around the world, we will increasingly see fresh water threatened by upland terrestrial practices. In our bioregion, particularly soluble reactive phosphorus, while it's a macronutrient for plant growth, when in excess in water, creates a situation called eutrophication, which facilitates the growth of cyanobacteria blooms. And basically this decreases the oxygen levels in the water column, and that can malaffect human and animal health, as well as decrease recreational opportunities. So ecological engineering solutions can be applied to landscapes that intercept that phosphorus and recycle it up. And when I'm talking about ecological engineering practices, I'm referring to associations between microbes, fungi, and plants. And one of the inspirations for me going back into academia to get the second master's degree is because I run a business called MycoEvolve, Ecological Resilience from the Soil Up through earthworks, education, and research. And my attempt to support land tenders, particularly farmers, in practices that mitigate phosphorus pollution, it seemed necessary to actually gather data on that. So this is an article that Dr. Joseph Borges and I published last year. It is a robust literature review on how mycorrhizae can be applied to protect water quality through supporting plant uptake and decreased leaching of phosphorus. And the reason we felt it was important to sort of establish this in the literature was formerly I had only seen mycorrhizae's phosphorus acquiring characteristics applied to biomass and crop production. So now that this is kind of in the literature, we felt it was necessary to gather more mesocosm and field data. So our third objective of pollinator habitat is because we're living in the 6th grade extinction. Insect decline is alarming, and biologist E.O. Wilson of recent memory warned us how invertebrates are foundational to trophic web, which if in peril can lead to ecological collapse, which I believe we are at today. As you can see from this diagram here of these different figures, these are some of the orders of monitored populations that are in decline. So where can we on farms support rematuration, protect water quality, and support pollinator habitat? Vegetative buffer strips. Now, the way these work is phosphorus can be retained in the buffer through particulate phosphorus settling and dissolved phosphorus being uptaken by bacteria, fungi, and plants. However, they're only temporary because that particulate phosphorus can remobilize in large storm events. In the fall, when we have senescence and all those plants die, that phosphorus gets re-released back into the soil and the water, and desorption from the soil matrix also releases that phosphorus. Riparian buffers do not retain 100% of the phosphorus load, particularly in areas where there's low plant coverage, such as buckthorn stands, because there's not that plant structure to catch that phosphorus and that plant structure to uptake that phosphorus. Similarly, these soils tend to oversaturate. And so you have a situation where these riparian areas over time become sources, and that aging perennial vegetation decreases in its water quality efficacy. As scientists, we measure this by soil, water, and plant phosphorus data.
Yes, it is.
The good news is that harvesting perennial buffer vegetation has been suggested as a way to improve saturation capacity and buffer longevity. Up to 5 to 45 kilograms of phosphorus per hectare can be removed depending on what species and how dense you plant them. Introducing the superstars of today's show, though I will say they are no silver bullet. Mycorrhizae fungi, here they're categorized in 7 groups based on their morphology, who they partner with, and where they grow. I'm only focused today on our buscular mycorrhizae. You will see me referenced as AMF, and ectomycorrhizae, you will see me referenced as ECM, but because I'm focused on farm ecosystems, I will particularly be focused on AMF. While this is a neat, tidy chart, I just want to mention that in terms of biologic and taxonomic reality, it's way more complex. The backdrop of today's research is really in the phosphorus cycle. And unlike carbon and nitrogen, phosphorus does not have an active gas phase, and the cycle is quite slow. There are these different pools of phosphorus. And what this diagram demonstrates, if you focus on the red lines, is where mycorrhizae can facilitate the movement of phosphorus between these different pools. So for example, releasing enzyme exudates in the form of organic acids, it can loosen up some of that phosphorus that's absorbed the soil matrix, and similarly facilitate weathering and dissolution, as well as stimulate microbes, and again, mineralize that phosphorus. And that moves phosphorus into a soluble form. You will hear me speak today of soluble reactive phosphorus, or SRP. And that soluble reactive form of phosphorus AMF contributes up to 90% of plant uptake. Plants are able to uptake that form. And similarly, there's decreased leaching up to 150 grams of phosphorus per hectare per year. This is particularly helpful in nutrient-poor ecosystems. It's an elegant relationship between the plant and the fungi. The plant practicing photosynthesis, harvesting sunlight, carbon dioxide, and water, feeds the sugars to the fungi. Meanwhile, the fungi expand the root zone, the rhizosphere, to access formerly inaccessible water, micronutrients, and macronutrients such as nitrogen and phosphorus, feeding that to the plant. Now, this demonstrates a more mechanistic view, which is a bit complex, but it's worth noting that the microbial community is a key player in this. And that's because if you go back into geologic time, microbes show up 3.5 billion years ago. And they have quite an extensive network by the time fungi show up, which is only 1 billion years ago. And then those two organisms have both between themselves and among themselves intricate networks by the time plants show up, which is only 500 million years ago. This diagram demonstrates the mycorrhizal communities in terms of their multi-species network and a concept known as host specificity. So if you look starting at the far left, this species in the Pollinator family partners with ECM, ectomycorrhizae. They often have fruiting bodies. And you see here's another species of Pollinator with purple with a different type of ectomycorrhizae and some cross between. Here you see some herbaceous species that have these little green endomycorrhizae, and you see there's not really an interaction between the two. Here are some other herbaceous species that also have endomycorrhizae that are partnering with this maple because maple is an endomycorrhizal species. So you see they're nested, they're asymmetrical interactions, they organize around a core of interactions, and this Orchidaceae over here is more modular. It's kind of partnering with its own particular group. So succession occurs, this will look different from year 2 to 3, and there's a resource allocation going on. Now, in mycorrhizal scientist circles, there's a lot of debate. For example, Dr. Susan Simard, you likely heard of from the mother tree. She has a more phytocentric view that the plants are determining this, whereas Dr. Tom Horton, a mycologist, has a more mycocentric view that the fungi are determining this. I think the jury is still out on that, but it's worth noting that ecologists often use a biological market analogy. Oftentimes, we have learned recently the ways that these taxonomical relationships can connect to ecosystem functioning, and particularly because of the last 30 years of molecular sequencing. And we also have a lot more questions about those relationships. What we do know is in terms of the soil, plant, and ecosystemic level, mycorrhizae fungi facilitate microbial activity and community composition. They facilitate soil aggregation through releasing glycoproteins such as glomus. They can facilitate chemical and nutrient cycling, water balance, and nutrient loss prevention. In the plant realm, they facilitate seed establishment, growth, community composition, heavy metal tolerance, drought resistance, nutrient uptake, and can protect against pathogens. And on the ecosystemic level, again, you see they contribute to community composition, nutrient cycling, water balance, and stability. Integrated ecosystems, you will often see that mycorrhizal communities are missing because their plant hosts have been removed either from drought or mining or agriculture or fire. Today, I'm going to specifically be focused on early successional sites, intensively managed plow fields, land laden with excess nutrients, and non-native species presence. It's worth noting that health can be restored to these ecosystems when you reintroduce mycorrhizae. And I emphasize the word conscious because it needs to be in relation to the microbial community in a symbiotic manner. So when I attempt to restore health to degraded ecosystems, I go back as far in time as I can. In this particular case, it brings me to the last glaciation, which is indicated here in this Abenaki map, the Great White Bear. Research indicates that as the glaciers retreated, the Abenaki ancestors moved their seasonal hunting and fishing camps back northeast, and they lived in these areas along the shorelines of what was glacial Lake Vermont. Then it became Champlain Sea, and then a bigger version of today's Lake Champlain, known as Lake Bituabga by the Abenaki. And during this time, not only did the climate change, but the vegetation shifted. Now, according to the fossil record, arbuscular mycorrhizae fungi have been on Earth for 407 million years. This was found in the fossil record in Rhine Shirt, and they actually assisted plants moving from water to land. But on this continental plate from Vermont pollen core studies, we have evidence that they've been here from the beginning of the last interglacial period when succession moved from boreal to hardwood forests, because we know these are mycorrhizal communities. As the Abenaki evolved from hunter-gatherer lifestyles to what permaculturists aspire to today through polyculture cropping, agroforestry, and seven sister mound planting, there were extensive trade networks. And I like to look at this map because this is a map of a time when the Abenaki were living on the land, and it indicates a potentially unfragmented network both below and above ground, and imagining that the water quality was quite high. So you will hear me refer to this today as a reference condition. Before I go through the atrocious trajectory of colonial history, I invite you to consider how these events have affected not only the phosphorus concentrations in the soil, but also the mycorrhizal populations. So we have early 17th century, the 90% of the Wabanaki are killed likely through smallpox, forcibly removed from the land. 18th century colonizer land practices replacing them, and this involves clearing for pasture and cropland, putting in transportation, tilling the soil, grazing, growing monocultures. And then the 1840s, we have the sheep craze. In the 1840s and 50s with railroads, you can now refrigerate dairy products. And so that increased herd and flock size. Then in the 19th and 20th centuries, you have roads and ditches installed without necessary vegetation. And then Shelburne Farms was able to consolidate 1,200 acres through a Vanderbilt legacy. Now, to their credit, they deemed this a model farm to experiment with latest agriculture and scientific practices. We're very grateful to this. It was deemed a historic landmark in 2001. And thanks, to the Webb family, they had a significant amount of wealth, so we're able to do this. And yet, it was difficult to hold on to this piece of land. So they certainly weren't going to say no to government subsidies that they and many farms throughout the country were offered to apply superphosphate fertilizer to the soil, a technology coming out of World War II. And then we have the Dutch elm disease coming in and non-native species replacing them. So if you put together these events, you have removal of a lot of the hosts that host mycorrhizae fungi, and the superphosphate fertilizer research indicates high phosphorus concentrations inhibit mycorrhizal populations, as do chemicals released from non-native species. And you also have a lot of animals releasing manure on land, as well as superphosphate, which is going to definitely increase the phosphorus concentrations in the soil, leading to a situation we call legacy phosphorus. So this brings us to our study site. This is Orchard Cove, which is known as a hotspot for both phosphorus and E. coli. I'm only going to focus on phosphorus today. The parent material is glaciolacustrean silty clay soils. They're highly erodible, but they're considered a farmland of statewide importance when they're drained. This area that I'm focused on is been under pasture for several decades, and it's surrounded by these two drainage ditches, and underneath is a tile network, which we do not have any maps for. It's worth noting that several students before me have gathered data on this site, many sites at Shelburne Farms, and written master theses on it. And I guess I just want to give you a metric of what concentrations we're talking about. So as recently as fall 2021, we gathered 6 samples of water leaving this tributary, entering the lake where the thumbtack was, and the mean concentration was 0.018 milligrams per liter. Now, Vermont water quality standards state no water leaving a tributary should exceed 0.01. So that's 18 times more. So we have a very modified, fragmented landscape, and on top of that, we have this 50 cubic yard pile of compost on a hill. All the topography leads to this drainage way. And I think it's just, again, worth mentioning that several students and I put together a plan, a final report that was approved by a UVM extension staff member that offered a vegetative treatment system to put below this compost pile, as well as recommendations to move it. The farm is not amenable to that at this time. And so this project is a little bit like a Greek myth of Sisyphus pushing the rock up the hill, because the actual source of phosphorus is not being shut off. So the best we can do is sort of compare our treatment areas to the control. And I just again want to emphasize that the vegetation in this buffer is primarily Rhamnus cathartica buckthorn. So here we are at Orchard Cove, and we're investigating how to reduce legacy phosphorus by restoring riparian areas dominated by buckthorn to a plant community that existed around the time of the Wabanaki Renaissance. And our research questions are involving Mycorrhizae's efficacy to improve legacy phosphorus mitigation, increase harvestable phosphorus amounts, and support a diverse pollinator plant community. So based on the literature, our hypotheses were that all forms of phosphorus would decrease, that corresponding plant uptake would increase, and that the restored plant community stability amidst diversity would ensue because of mycorrhizal presence. So this area between the reds is our installation area and is known as the interception of a critical source area between that phosphorus and the waterway. We put in 3 treatments, and yes, I know scientists in the room are saying that's pseudo-replication, and you're right. It was the best we can do. We did do true replication in the greenhouse. And you'll see the OIV is original invasive. These are buckthorn control that we kept in place. And the difference between the two RVs, RVM, if you think M. mycorrhizae, the RVM is inoculated with mycorrhizae. One of the reasons we left the control between was in order to prevent the mycorrhizae from growing into the uninoculated plot. So this is one of a series of concept maps that demonstrates the sort of multi-synusium, which refers to the multi-levels in the forest and the plant communities. And this was informed both by advice from Dr. Annie White, but also intuitively, I'd been walking and paddling riparian areas and looking at what natural plant communities grow together. So here we have the first criteria, if you recall from that early slide, of what deems a site restored. All of these species were around during the Wabanaki Renaissance time. Functional groups are present, and they can endure these high phosphorus soil conditions, as well as hydrologic soil group D, which is clay, is in this case, and quite wet. And so if you look at this plant palette, you see at least 3 notable features. First, in the mycorrhizal category, these species are either AMF or ECM. Some, such as in the Salicaceae family, are both. You see a flowering window from February to November, and that's really important for providing what's called flower constancy so that these invertebrates can go through their entire life cycle. Now, obviously in February in Vermont, you're just gonna have catkins on elm and berries on the winterberry. And then the third notable feature is the Wabanaki uses column. And 88% of the species that we work with have either edible, artisanal, medicinal, ceremonial, or utilitarian purposes for the Wabanaki. And this is very important to us because one of our objectives is over time to allow these restoration research plots to also become Wabanaki harvest ways. This is what the plot looked like in fall 2019, the buffer, when I first started this research. It was very intimidating, all of this buckthorn. And I just want to give deep thanks to Mike Bald of Gottweeds, who's been a mentor to me in how to remove this buckthorn in ways that do not malaffect the water and the soil. In the summer, looking at it, again, you can see it's 90 to 95% buckthorn. The other 9 to 11 species it partners with are honeysuckle, poison ivy, horsetail, several sedges and mosses. And Doug Talamay, an entomologist and indirect mentor of mine, talks about the importance of when you restore pollinator habitat, you want to bring in ancient plant lineages that have millennial long relationship where the insects can obtain their life cycle needs through these plants. And a lot of these non-native species have only been here for a century and a half, and therefore a lot of the insects don't recognize them or can't obtain what they need. And doing so without chemicals is really important to us because currently 42.3% of ecological restoration projects rely on herbicides. So what Mike Bald taught us was that if you cut the stumps at belt height and then strip off any regrowth 3 times in 2 seasons, you get a 90% death rate. And we saw that again and again. So looking right here, standing here looking this way, a year later, this is the waterway, all surrounded by native pollinator species. And look who shows up on those stumps. Trametes versicolor, one of my favorite saprophytic mycofiltrating fungi. So I'm just going to talk through the installation. Using a low phosphorus soil, we were able to pasteurize this. and inoculate half with a commercial endo-ecto mycorrhizal mix. We then potted up the plant's bare root at Vermont Wetland Supply in Orwell. Again, only half of them inoculated, half in uninoculated, and also inoculated half the riparian seed that was all native seed, and then let them sit for 6 weeks. Now, it's worth noting that this is when COVID hit and all research at UVM shut down. And I was so grateful that we wrote MycoEvolve into the grants so we could keep going. And I am incredibly grateful to the grassroots community I'm part of, because without them, the preparation of the site would not have happened. So we had, this is what the site looked like in March 2020. Thanks to Shelburne Farms staff, they actually cut all those buckthorn at belt height. The trees you see standing here are any ash or maple. We vowed to not remove any of the native species. And then we had 12 COVID compliant, physically distant work parties, and basically removing all the stumps away from the waterway. And some of them were huge. And then wheelbarrow them to a pile where they were chipped. We also put fences up around the 2 restored plots to decrease herbivory. And we flagged all the native plants. So you can see this is what one of the restored plots looked like after we removed all the buckthorn, except you see we left all the stumps by the waterway. And I was really grateful we were able to hire Vermont Youth Conservation Corps. I used to lead crews for them, so it was a nice full circle. Here they are on installation day, laying down jute erosion control mat over the seed we planted, and then we cut into that to plant so that that held the soil in place till the plant's established. And this is sort of a view of it on installation day, one of the restored plots. And this is a view of it a year later. So for maintenance, yes, we did experience a very dry summer 2020, if folks remember. So we're really grateful to Shelburne Farms. We're filling this truck up every week, hauling it down. And then through hoses, I watered both restoration plots. And luckily 2021 was more wet. We didn't need to do it as much. However, the grasses really took off spring 2021. So we had to hand scythe them. And you can see the shrubs and trees starting to rise up. And here was after the second scything, the trees and shrubs could really establish themselves. So here we have the next 5 criteria. The system is functioning according to its developmental phase. It's integrated with the surrounding landscape matrix between field and forest, and potential threats to the restored system are eliminated. Yes, I did need to pull up a lot of baby buckthorns. I did need to do the strip back on the stumps, but it wasn't too much. Over time, the system demonstrated resilience. There was less maintenance needed, and definitely indicated that it is evolving. And so in terms of data, for phosphorus, we sunk these lysimeters, which are ground wells, 6 of them in each of the 2 restored plots by willows, and in similarly sized by buckthorn in the control plots. And when there were storms greater than 0.5 inches, we would pull up that water and filter it and test it. For water extractable phosphorus, we actually gathered soil in all of the treatment plots. And then through a series of agitations, we're able to pull out that soluble reactive phosphorus, as well as analyze the total phosphorus of the soil. In terms of plant biomass, this is interesting because originally I was following coppicing recommendations, which say to harvest when you prune fruit trees, which is the end of winter. However, our mesocosm studies indicated at this time, most of the phosphorus is in the roots at this time. Not a great time to actually physically remove phosphorus. So this is me actually pruning, coppicing. If you're not familiar with coppicing, it's when you're removing the above ground biomass. Since they're perennials, they'll regrow. Doing this in September, enough time so they could harden off before the equinox. And you will see from the data, there's statistically significant more phosphorus in the plants at this time. So then they were dried, and they were, this is kind of what the leaf looks like. After drying, we ground them in 2 machines, thanks to the Hort Farm for the older, rougher machine, and then the grain lab next to us for this machine. Here's one of my interns, Mary Robideau, putting them in containers. And in terms of pollinator habitat, so what we did was we put 4 transects parallel to the waterway using random numbers every month of the growing season in all 3 treatment plots, recording every species that we saw. In terms of plant species diversity, I put 5 of these quadrants in each of the 3 treatment areas, again, using random numbers every month of the growing season in all 3 treatments. And not only recorded what species were there, but how many of each. Today, I will only be sharing about my plant species richness data. Mycorrhizae was not an objective, and I chose to gather the data anyway because I felt it was important to check our assumptions. Did our study site actually get set up the way we intended it to? And so obviously I wasn't going to dig up the plants. Normally that's what people would do, is you actually look at the colonization in the roots. And for example, this is a arbuscle, which is a morphological feature that you can tell about colonization. Instead, I focused on what's called the extra-radical hyphae, which grow out into the soil. And the way we did this was by gathering soil in each of the 3 treatment areas. Here is another intern, Paige Sterling, through soil extraction, and then putting them on these microscope slides with these grid line stickers and creating a modified grid line intersection method where we created criteria to differentiate the mycorrhizae fungi from saprophytic fungi and dark septate endophytes. So looking at this data, you see a few interesting things. These on the y-axis are the counts, and these are the 2 years. So the pattern is that, yes, indeed, our study site was set up the way we intended. The restored vegetative mycorrhizal had the most mycorrhizae fungi. Great. What was surprising was that buckthorn actually have mycorrhizal populations. I didn't know that. And it's also interesting to know that they release a phytotoxin emodin that reduces other plant and mycorrhizal communities. So that's why they may only have 9 to 11 plants they partner with. Now you may wonder, why are there mycorrhizae in the restored vegetative? That's likely because of the native plants we left in place. So it's worth noting that we are really looking forward to doing some DNA analysis on the taxonomic groupings of the mycorrhizal communities in the buckthorn compared to the restored mycorrhizal communities, because it's going to be very interesting, not only below ground, but also above ground, to look at those community composition differences. Now, the most notable phosphorus data that we found was from our water extractable phosphorus. Looking at the diagram on the top of the page, the x-axis is time, so we have July and August, and the y-axis is soluble reactive phosphorus concentrations. And you can see a pattern here. So while there's no statistically significant difference between treatments, the trend is that the uninoculated restored plots, the red, has the most soluble reactive phosphorus, followed by the buckthorn, and the least is in the inoculated mycorrhizal plot. Now, if you do the data on this in terms of statistics for hyphal density, as you'll see in the lower diagram, the x-axis is hyphal density, the y-axis is soluble reactive phosphorus concentrations. As there's more mycorrhizal hyphae, the soluble reactive phosphorus concentrations go down. So this could be because the mycorrhizae are doing what they say in the literature. They're moving that soil, that phosphorus out of the soil, out of the water into the plant. But it could also be due to spatial variability. So more research needs to be done. In terms of plant phosphorus uptake, I did mention that there was much more phosphorus removed in September compared to April. Now you'll see between the 2 restored plots, there really wasn't a difference. There was the same trend, but it was worth noting 3 times more in the end of the summer. And so we really believe that coppicing timing is a really critical element. And in fact, this could be suggested as a best management practice to improve riparian buffer management for water quality protection. And I emphasize cyclical coppicing for 3 reasons. One is that research indicates that phosphorus accumulates in a maximum amount between 3 to 4 years. So you're not going to do it every year. 2, the structure of those plants is really important to provide overwintering habitat for invertebrates, early and late season migrants. You're not going to want to, plow it all down. And also we talked about that plant structure catching particulate phosphorus. So again, you want to leave some of that structure in place. So cyclically coppicing is a way to meet those objectives. Looking at our plant habitat richness, you see a few things. So the 2 restored plots, the red and the green, are pretty similar. But it's worth noting that 1.7 more species showed up than we planted. And this is significant because it indicates that some sort of autogenic repair, some sort of succession beyond the human, ideally the human gets out of the way after you restore an ecosystem, starts to happen. And as expected, there's 4 times more species in the restored plots than the buckthorn. So it definitely will be neat to track this over time. There are many more species besides willow that can be used to remove phosphorus. For example, elderberry. And folks, if you want some elderberry syrup, there's some over there. People are probably familiar with it, as a great immune tonic, as well as a great food that not only many Vermonters and people throughout the bioregion, but also Abenaki use. It actually removes 3,598 milligrams of phosphorus per kilogram of dry mass. And there's evidence that it can provide economic return for farmers. So this is just indicating that there are a lot of other species that can also remove phosphorus. More research needs to be done. And here you see that 10th criteria of a system satisfying a mitigation function. So here, removing those elderberries, coppicing those willows, you're physically removing that phosphorus from the landscape before it goes back into the soil and water if you left it in place in the fall. There were many limitations to the study. So first of all, due to time and budget, it was only a master's, not a PhD. So there were many questions we weren't able to answer. Pseudoreplication is never recommended. Commercial versus local inoculant. So you can ask me more about this in the question answer period, but suffice it to say that there were several good generalists that partnered with our species, and then there were some that just didn't in the commercial inoculant. There were several covariants of shade and unknown tile drain outlets that may have affected our data. And there's a lot we don't know about host plant fungal relationships beyond this AM-F ECM specificity. We were not able to gather quantitative data on actual pollinator visitation. I will say, while doing my plant surveys, I saw many carpenter, mason, and sweat bees, as well as bees and flies, beetles and flies. But if folks have the skill set and want to join our team, we'd be super grateful. And we're particularly interested in looking at this between restored, inoculated, and uninoculated plots, and then pooling that data and comparing it to the buckthorn control. In the research, there is a known lag time of phosphorus mitigation of 2 to 10 decades. And that's actually if the source of phosphorus is removed, which it's not in this case. So we sort of have an infinite lag time in this particular case. And our project does not address equity issues of land access and ecoliteracy for our Black, Brown, and Latinx sibling communities, though we do intend to in the future through employment and educational opportunities. So going back to this originally stated first objective, which was an objective that evolved, we believe that our project can serve as a model of ways to kind of transform the way we work with landscapes, that we can not only mitigate pollution, restore pollinator habitats, support farmers' economics, but also allow them to become harvest ways for Abenaki. I'm grateful to say that Chief Don Stevens has a longstanding relationship with Shelburne Farms, and this summer we're aiming to craft a memorandum with Shelburne Farms to turn these into not only research sites, but also Abenaki harvest ways. And Vera Longto of the Abenaki Artists Association is inviting us to sort of write a letter of invitation of harvest so that Abenaki, after we gather the data we need, can come and remove some of those 88% of species that are helpful to their peoples. And I think I just want to emphasize the word gesture here, because you can't remediate ecological or social devastation in a 2-year project of like 400 years of devastation. So it's just a gesture. But the idea is that by the time Bina is our age, at least the needle will have moved a little. And this is part of a much greater rematriation movement around the world, particularly in the Northeast, coming out of the Seeds of the Renewal, which has become the Abenaki Land Link Project, state parks erecting signs with place names. And here you have our attempt to address that criteria of social injustice inherent in the environmental damage also needing to be repaired. Going back to our original research questions, do mycorrhizae improve legacy phosphorus mitigation? Too early to tell. Do mycorrhizae increase harvestable phosphorus amounts? This is a complicated question. It depends on concentration in the biomass. We really didn't see a difference between the 2 restored plots at this point in time. Harvestable biomass, we're not going to harvest all those plants yet, so we don't really know. Timing, yes, definitely 3 times more in late summer than early spring. Do mycorrhizae support a diverse pollinator plant community? Both restored plots seem to, but 2 years is a really short time. There really didn't seem to be a difference between the inoculated and uninoculated. So the fact that 1.7 more plant species showed up in both of them indicates some sort of succession is happening, but we need many more years of data to be able to tell the difference between those 2 restored plots. So recommendations on 2 years of field and mesocosm studies are gather site history from pre-colonial and local indigenous knowledge, remove non-native species with a 3-cut and 2-season approach, design multi-species site-specific native polycultures with local indigenous peoples access, guidance, and use in mind, inoculate plantings with native soil without disturbing the soil, cyclically coppice late summer for pea removal, halt upland phosphorus contributions such as compost piles or amendments. And if you can't do that, at least put in some forms of green infrastructure that intercept that phosphorus and uptake it, knowing that whatever phosphorus is entering that needs to be less than the amount you can remove through coppicing. So I just want to acknowledge I'm standing on the shoulders of so many giants. This is really a community project. I'd like to just start with my advisor, Dr. Joseph Gores, who has not only mentored me very patiently into a research scientist, but also really done a tremendous amount to support the research. I'd like to give thanks to the Hurley Faulkner Lab, who I've learned so much from being a part of, as well as my committee, Dr. Terry Delaney and Dr. Stephanie Hurley and Dr. Joseph Gores. There are many of Bonneke collaborators that have mentored me, and grateful to continue working with them. Several professors at UVM have been incredible mentors and teachers to me, as well as staff members at UVM who helped me access a lot of information. Many water quality professionals and other farmers have helped. I want to give thanks to all the funders, UVM Plant Soil Science, UVM College of Agriculture, Life and Science, the Lintelock Foundation, the UVM Gund Institute for the Environment, Northeast SARE, New England Grassroots Environmental Fund, grateful to collaboration for Vermont Youth Conservation Corps. I'm also grateful to collaboration with Shelburne Farms, and particularly the farm manager, Dana Bishop. And there's a lot of unacknowledged support, such as the microbes, fungi, and plants who are doing most of the work. And I think in conclusion, and my interns, there's so many more people to mention, grassroots community, anyone I haven't mentioned, thank you. And I just want to dedicate this work, if it has any merit, to the earth, to my ancestors, and to the future generations, as you see here, these homeschool students who helped in the site preparation for their future as well. So thank you for your time. And I guess I can take questions.
There are some.
Questions?
Yeah. Why not?
Well, I got a whole bunch. First of all, thank you very much, and it's very relevant to work that we are doing in the same geographic area, dealing with buckthorn and trying to reestablish native plant communities. And like I said, I can go on and on. I'll just one thing that we are doing, and I don't know if you looked at this, we definitely see throughout the Champlain Valley, you have incision, in all of our streams, and generally a lowering of water table. And what I think is happening is you lower the water table, you lose your wetland hydrology, you lose your wetland plants, and all these woody invasives come in. And so one of the things that we're doing through stream restoration processes is trying to restore hydrology, and then cutting the buckthorn. They will be growing in a less, in a harsher condition for them. And then hopefully that will invite wetland plants coming in. But one specific question I have for you, when you were looking at the phosphorus remediation, so you had a system where you had sort of a continuous flow of phosphorus in, right, from the compost piles?
Well, I don't know how consistent it is. The hydrology at that site is really complicated. So there's definitely, overland flow from storms. There's definitely subsurface flow, but there are tile drains that may be bypassing the buffer. Right. And then, there is certainly phosphorus continually coming not only from the field of legacy phosphorus, but the pile.
Right. So to really capture the phosphorus remediation figures, is soil reactive phosphorus the one that you want to monitor, or is there an overall phosphorus number?
Okay, that's a great question. I think that several metrics are a good tool. The one that I actually found to be, soluble reactive phosphorus is the form that directly creates cyanobacteria blooms, and it's also the one that's uptaken by plants. But I do think looking at total phosphorus concentrations over time, while it will change slower, is an important endeavor. I do just want to reference, I have to kind of show you this one slide of a tool that we discovered called phosphorus saturation ratio. We used in our mesocosm studies, and it's a very useful tool because what it does is it, based on your soil texture, if you do a Malick test and you get the phosphorus concentrations, which will also show you the ion and the aluminum concentrations, and you do that formula on the top right, based on your soil texture, you will find a critical level that above that, it's more likely to leach, and below that, it's less likely to leach. So I think that's a really valuable metric also. But I definitely would say a few metrics, not just one. And research indicates not only seasonally, but through the years, because it's such a slow moving cycle.
Got it. Thank you.
Yeah.
Excuse me, sir. You said we, but I don't know who you are, who you're representing.
Oh, well.
Interested to know.
Yeah.
Who am I representing? I have a business, we provide a number of field services in the name of ecological restoration, and that's Riverscape Ecology.
Oh, neat.
Right.
Cool.
So if anybody has buckthorn IV skills and chainsaw skills and wants some work, give me a call.
Yeah, I should introduce you to Mike Ball, who got weeds, because he can
Yeah, no,
train
I know
crews.
Mike.
Okay,
I know
yeah,
Mike, yeah.
great. Any other questions, comments? Yes.
I
Doreen.
have a million questions.
Another
as well.
mycorrhizal scientist in the room.
And they're all mycorrhizal related. So I'll go with the most relevant
Okay.
first in my mind. I know that you're interested in doing molecular work here, which would be awesome to do. Really annoying to do, but really awesome to do.
Yeah, next steps.
If that goes through and you get a sense of the community composition, not only the mycorrhizal fungi, but you have information on the above ground plant community, would you hope or would you expect to uncover specific pairings of fungus and plant that might be more adept at removing phosphorus? I guess the question being, there's this continuum of function between different species of fungus and plants. So would you expect to see specific pairings that could be really helpful in this specific case?
That's a great question. I mean, maybe if I'm lucky by the time I'm 70, I'll get there because I'm aware, like even Sandra, who's here, has been helping me learn about DNA, and I know it's going to be a steep learning curve. But my understanding is there's a couple steps that need to happen. First, I have to learn DNA with AMF, which is a nightmare in itself, I've heard, in terms of heterogeneity. So that's one thing, is being able to get that down. I'm expecting will take a few years. And then also the phosphorus accumulation potential of these plants, which is something we have written grants on to now expand our quiver of that and hopefully get a handle on. So simultaneously, we'll be working on both of those. So figuring out which plants can take up more phosphorus, and then being able to identify their taxonomic mycorrhizal groupings. And then obviously all those riparian practitioners and farmers who keep asking for lists, hopefully one day something can be delivered. But I think that my humility has increased in realizing it might be something I hand to the next generation and just move it a little and they'll get there.
For sure.
Yeah.
Good points. Yeah,
Yeah.
It's definitely been a trouble in my own work because we don't know anything about the community composition of these fungi. And we just assume that just throwing a bunch into a place will have a benefit, but we don't know who's doing what.
Yeah.
So that's definitely helpful. One other question I had was with local inoculum, which, as you know, people think that's a better alternative than commercial inoculum at this point. Is there a concern of introducing root pathogens in addition to introducing helpful fungi to a system?
That's a great important point. And actually, we have a project starting this spring at the Intervale where we're going to be using local inoculums. Another thing I have to learn in the next month is how to do that. And the buffers that we are going to be taking the soil from, I was curious about finding out the best we can beforehand about root pathogens, because I've seen in the literature that can be a problem. And I guess that's kind of that statement, right, where you move one thing and it could have a whole nother cascade. So if you could do an assay beforehand and you had time, maybe in the winter, that would be ideal for sure. That's a great question.
That's all I have for right now.
All
I'll
right,
find you
thanks.
later.
Yes.
Other questions? We have only a couple more minutes before we have to
Oh, yeah.
get to the more formal part of this defense.
Yes, Sana.
Thank you so much, Jess. That was incredible. Congratulations. I learned so much. Lots of things that I've literally never heard of before. So it's really impressive to see you with such confidence and grasp of this material. It's incredible. And my question obviously comes from someone who's not an expert in any of this area, but I see how relevant things like the payments for ecosystem services from the Vermont Department of Ag could be, and your work is, I think, incredibly important in that space. And just wondering what future plans do you have in working with the Department of Ag and getting some of this rich research in their hands.
Thanks. Thanks for that. It's a great question. I guess, you know, I did participate in Dr. Eric Roy's lab for a little while where we discussed payment services for ecosystem services, and I appreciate his sort of kind of potentially cynical lens that being careful of people monetizing these, and you know, and so I think I'm going to be very wary of that. I kind of want to keep my head more buried in the humbling science of this and building relationships with the Aboniki and kind of doing that work. I think I'm nervous about the whole economic market part, just because capitalizing on it, greenwashing already, as one of our mentors, Tom Horton, calls about these, you know, mycorrhizal commercial inoculants, snake oil, you know, people capitalize on that, throw whatever in there. And so I'm nervous about that. I appreciate the importance of how economics speaks in our system and it has power. And I'm sure people doing really good, honest work on that. If I find and connect with some of them, I'm happy to collaborate, but I certainly want to, I think the science is going to keep me pretty busy and growing with the Aboniki, because it's a little dicey, I think, some of the challenging parts of that.
If I can chime in there really quickly, I mean, being able to make some of the ecosystem service available to Aboniki is an economic act, right? It's just not in the system, so to speak. You know, it's a bypassing economic systems.
Right, reworking that capitalist market paradigm of other forms of capital.
Rhonda.
Thanks, Todd.
Thanks.
So I was wondering, to go back to this idea of reconciliation, you know, so right now you have a snapshot that you can see based on kind of, you know, a year or two out from your restoration or whatever your kind of major transition in this local spot, you know, in terms of what the community looks like. How do you think you can kind of project forward as to whether it will revert back to kind of this buckthorn dominated ecosystem? And, you know, what's your thought as to like, what are the ways to kind of protect against that versus is it just going to be inevitable?
Are you referring specifically to the restored plots that are surrounded by buckthorn on either side?
Yes.
Great question. Well, one option, Dana Bishop said, hey, could you do this to the whole, you know, how much would that cost? So maybe we will write a grant and do that at some point, which would be awesome. But if that wasn't the case, I do think it will require, you know, maintenance. Usually these systems, the first bunch of years require a lot, but I do think over time they will hold their own. That may be an overconfident statement, but my understanding is that there is some sort of, based on mycorrhizal communities, I think over time that there could be some sort of, I don't know if understanding is the right word, but some sort of boundaries that over time can be created. It might take like a decade still of maintenance and monitoring before they'll hold their own. But I think that over time they will hold their own. The tricky thing is that some of those buckthorn have berries hanging over our plots that we continually need to prove. And they have like an 85% germination rate. So maybe it's more than a decade of maintenance. It might just be one of those things that you need to continually maintain, but definitely a good thing to think about. Appreciate that question.
Yeah, just to follow up, it just seems like there might have been certain kind of community kind of attributes that allowed the invasion to happen in the first place.
Oh,
And
yes.
just kind of to think about whether these plots have moved to some other state or whatever to kind of prevent that same process.
Yeah, well, on that level, if you think subsoil, the mycorrhizal communities and the enzyme exudase they do release, I do think actually that some of those chemicals, at least like Paul Stamets' work, indicate that there's certain sort of myco buffers that can be created. So for example, you can create a myco buffer around a particular ecosystem through mycelium that will prevent certain species from coming in. I definitely don't have that knowledge level yet, but I could see over time if buckthorn release emodin and these other mycorrhizal, at least the generalists we use that did work, maybe over time would release some sort of chemicals that would create a buffer. Definitely like another whole steep learning curve, but that's a great thing to think about. Thank you.
Again, a great help. So I wonder about the, in like 1 second, why are they not, why is Shelburne Farms not willing to change management practices to stop the,
Yeah, that's a great,
great
great question.
Not to throw change it, Shelburne
Yeah,
Farms, but rather
under the
to
bus.
understand
No,
farmer decision
Yeah,
making.
for sure. You know, I've gently sort of asked And my like elementary understanding is that there's a lot of land that's managed by very few people. The farm manager has 2 young children and is managing a tremendous amount of land. And in terms of the topography, that landscape, like there's a sugar bush up here, there's a CSA market farm here, and then there's pasture. And it's very convenient in terms of like the bucket loader to go into this log landing where it is. So it definitely require like significant infrastructure shifts, but never too late to give up hope. Good question.
Okay, so I'd like to cut this off right now.
Okay, thank you
Because
so
we have
much,
the other-
everybody, for coming. And thanks, everybody. Thank you so much for
I'm
coming.
sure that if you have more questions, you can talk to her directly.
Very familiar.
I've been just on stage with-
So you're doing it with Blair.